Okay, let's go ahead and get started. Hopefully there are no fire alarms today. This semester has been nuts. We've had so many crazy things happen, polar vortex, this, that, and the other, so we're just going to try and make the best of it and get through the material as best we can. I might skip a few slides to make up some time, and obviously I'll let you know that, and if I'm skipping it, you're not responsible for it, so don't worry about it too much. Okay, so this is where we had left off. We were actually having a little bit of a Q&A about cancer. I know somebody had asked a question when the alarm went off. If that person wants to re-ask it, I'm happy to answer. Yes?
That was the double mastectomy with the cancer gene for breast cancer patients, or people who test for possibilities to use the BRCA.
BRCA1, yeah.
Whatever. Is that really only to test for breast cancer? Or is it, do they still run the risk of developing, how does that work?
So we'll actually cover BRCA1 and BRCA2 many slides later, but to answer your question, so breast cancer mastectomies, removal of the tissue is a very effective way to treat it if you catch it early enough. The tissue in the breast, as we'll cover later, it rapidly proliferates and throughout life for a variety of reasons. And so it being a proliferative tissue is at risk for cancer. BRCA1 and BRCA2 are two genes that are actually important for the DNA repair pathway, I believe, if I remember that correctly. We'll get to it. So mutations in that tissue tend to put you at higher risk of breast cancer. So the reason they screen for that mutation is because people have done enough studies to determine that if you have a mutation in this gene, your risk of cancer is so much higher than if you didn't have it, that it's worth the surgery, all the issues that come with it. It's worth doing that just as a safeguard because you are likely to develop that cancer as you get older in life. So it's just sort of like a cost-benefit analysis where it's like, okay, your odds are so high, and we all live quite long in general now, so let's just go ahead and get rid of that right now and avoid the possibility later. So that's preventative measure, exactly. Yeah. Okay, so that kind of concludes last time's general overview of cancer. And then these are the three main topics that we're going to cover today's lecture, next lecture. The first is mutation and proliferation, which you've already started to hear about. The idea that good cells go bad, that you start over-proliferating, that's the beginning of forming a tumor that then could become cancerous. Then we'll talk about angiogenesis, probably not until next class. And angiogenesis is the formation of new blood vessels, which is an important part of a lot of normal things that happen in you, especially when you're young and growing, but it can also be co-opted by cancer for negative consequences. And then the third thing we'll talk about is invasion and metastasis. How do you take that original tumor that's benign, or let's say is growing like crazy, and turn it into something harmful? What causes cells to migrate away and populate other tissues, like I mentioned to you last time, and then inhibit the function of different organs and actually cause problems for you? So that's going to be covered in invasion and metastasis. All right, so let's start with mutation and proliferation. And again, you heard a little bit about that already last lecture, or the 30 minutes we had, basically. So most cancer-causing agents, which are carcinogens, so cancer, carcinogen is an agent that causes cancer, are mutagens, cause changes in the nucleotide sequence of DNA. And I made that distinction last time in response to a question that not every mutagen is a carcinogen. A mutagen, which is anything that can mutate your DNA, is only considered a carcinogen, which is more of a almost clinical term, because it's been proven to cause cancer. Not every mutation leads to cancer, right? So there's a distinct difference there. Some examples of carcinogens are listed here. Various chemicals, not all chemicals, right? So, there's a lot of fear-mongering, unfortunately, on the internet these days about chemicals and long names. There's an individual who I'm going to paraphrase a quote that, if I can't understand the name on the ingredient list, then why would I eat it? Well, that's a ridiculous way to view things, right? If you can't read something, that means it's dangerous? No, of course not. H2O is dihydrogen monoxide, is a very classic example of this. Dihydrogen monoxide is not bad for you, it's water, right? So not everything that's a chemical or a molecule or something is bad for you, but of course there are things that are toxic to us, many things. So chemicals can be toxic in different ways, and one of the ways they can be toxic is in some cases by causing mutations that can lead to cancer. X-rays, I think you're all familiar with that. You can mutate DNA really rapidly with X-ray radiation. Sunlight, UV light, we should wear sunscreen for that reason, because you don't want to generate too many mutations in your basal layer of skin cells that can lead eventually to melanoma, skin cancer. And even viruses can get inside your DNA. One of the things viruses do is they often integrate into the DNA of the host cells, because that's a nice way, easy way for them to proliferate and make more of themselves. And sometimes in doing that, they might jump right into the middle of a gene and mutate that gene or something. And that can lead downstream to cancer, because you end up causing mutations that create problems like we've discussed. Bacteria can also have a similar effect sometimes. So as you can see, there's a wide range of influences that can be carcinogens. Okay, I'm going to show you a video, and it's perhaps, I don't know, it can be a slightly frustrating topic. Like, we talk about all these things that can cause cancer, and it's like, okay, great, if I don't smoke, if I put on my sunscreen, if I do this, if I do that, I'll be okay. And, well, that's only partially true. What you're doing is you're reducing your risk of cancer, but we all get old, we all get mutations, and as I think I mentioned last time, if you live to be 100 years old, you probably have some cancer growing inside of you somewhere. Whether or not you ever detect it is a different story. So there's only so much you can do, and that's because there's been a lot of work showing that actually you have random mutations, and that many cancers are just caused by random mistakes in DNA. Like, nothing you did, nothing you were exposed to. It's just going to be random mutations. Particularly as you get older and your machinery for fixing those mutations doesn't work as well because your cells are getting old. So I'll show you this video to kind of illustrate that point. Oh, and then if we have time, we'll come back to these fun ones. But let me, play this video for you.
Stories like these emphasize how important it is to understand what causes cancer.
Research over the past 30 years has led to the discovery of exactly what causes cancers.
Mutations cause cancers, but what causes these mutations? 3 factors are responsible. First, random errors occur every time a cell divides and copies its DNA to make 2 new cells. This routine process happens millions of times every day in everyone's body and usually does no harm. However, cancers can occur when such errors, simply through bad luck, affect an important gene rather than an unimportant gene. Our research suggests that nearly 2/3 of the mutations that cause cancer are due to these errors, this sort of bad luck. The second factor is environmental. Some people add to the number of mutations in their bodies by their behavior, such as sunbathing or smoking. The third factor is inherited. Some people are born with mutations that make them more likely to get cancer.
Let's use the analogy of typing using a keyboard. Because we are humans, the task of typing will of course result in typos. This is just like mutations that occur when normal cells divide. Behavior, such as typing while tired or when distracted, can cause even more typos, just as certain behaviors can cause extra mutations in DNA. A factory defect in the keyboard, like a stuck key or a missing key, can cause even more typos, just like an inherited factor can cause more mutations in DNA.
Let's look at pancreatic cancers as an important example of these principles. About 3/4 of the mutations that cause pancreatic cancers are due to errors made during the division of normal pancreas cells, that is, bad luck. About 20% of these mutations are due to environmental factors such as smoking, and the remaining 5% of the mutations that cause pancreatic cancer are inherited. Our research suggests that nearly 2/3 of the mutations that cause cancer are due to these errors, this sort of bad luck. Everyone should avoid behaviors that increase the chance that they'll get the mutations that cause cancers. But many people will develop cancer no matter how perfect their behaviors are. These people should certainly not feel guilty about getting the disease. There is nothing they could have done to avoid it. Because some cancers are inevitable, we believe that more research to find ways to detect cancers earlier while they're still curable is urgently needed.
Okay, so what I want to emphasize is the takeaway is not now you should all go out and smoke and eat whatever you want because, oh, most of this is going to happen no matter what I do. Because there's one important point that's not in that video, which is the timing of the cancer. So if you do smoke, you know, even though it's a smaller percentage of the cancers being due to those behaviors, if you smoke, for example, you're going to develop those those cancers you're more likely to develop them earlier. And obviously, if you're in your 40s, your 50s, your 60s, whatever, you want to live a life that's a healthy life. And as I mentioned, you know, you may eventually develop cancer just through bad luck, but you want to decrease the odds of that. And then hopefully by the time you're a really, really old person, and you're 100 years old or what have you, and you die of natural causes, it's not going to matter if you've had DNA repair-based cancers causing mutations. So yes, most cancers are due to DNA repair mutations that you can't do much about. But when you get a cancer, or if you're going to even know you ever had one, and the kind of life you have is dependent on your behaviors. So by all means, don't take this as I should start smoking or something. Definitely don't smoke. That's probably the number one thing you can do to decrease your risk here. All right. Yes?
Is there any correlation with living a healthier lifestyle and people are more likely to either notice their symptoms because they're healthier or they go to the doctor more often?
Oh,
Regular checkups or anything like that, and thus the cancer may be detected a little earlier?
That's an interesting question. So the question is there any correlation between leading a healthier lifestyle and or going to the doctor more often for checkups and detecting the cancer? So the second part of what you asked is well, so I certainly not my area of expertise in terms of has anybody done a correlation study? I'm sure people have. So the first part, leading a healthier lifestyle, I would guess is not directly relevant, but the second part, you mentioned going to your physician, absolutely. One of the important things about catching cancers is early detection. Now that doesn't mean that you go to your physician and they're always going to catch it, but if you're keeping tabs on how you're doing and you're quick to note anything that's highly unusual, if you start exhibiting some symptoms that might indicate, let's say, one of your organs isn't functioning well, whether it ends up being a cancer or something else, obviously you want to go get it checked out. And it's very true that there are populations and groups of folks who don't, for whatever reason, whether it's time, money, access, remembering to do it, who aren't going to physicians often and early enough to get checked out, and then those things aren't detected until a much later stage of life, and then it's a lot harder to deal with. As a very specific example of this, I have a collaborator who's a clinician here, and she was telling me about so neuroblastoma is a childhood cancer. Usually you get it when you're quite young, but she was telling me about a patient who was 30 years old, and it doesn't mean that the patient had the cancer at age 3, but that patient had so many other broadly speaking issues about seeing physicians and getting help that that person needed, that it's entirely unclear when that person showed up at age 30 when they first developed it, and by the time they showed up it was not possible to do much to help, right? So absolutely see your doctor if you feel like something's off. It helps. Yeah. Okay, the next thing I want to tell you about is an example of how viruses can cause cancer, in part because it integrates some of what you've already learned about a couple of different proteins. So first I'll kind of show you in the slide what's happening. The uterine cervix is a highly proliferative region that can also have cancer quite often. So it turns out that certain papillomaviruses are thought to give rise to this cancer. These papillomaviruses have double-stranded circular DNA, and in a benign infection, which is shown on this side, in other words, a case where you're not going to have a risk of cancer coming from it, this is what happens. So you've got this plasmid from the virus in red, and here's the host cell's chromosome, just an example. Obviously, going to be lots of chromosomes in there. And what's happening is that there's a bunch of proteins that the virus is making because it's hijacked the cell, like viruses tend to do, and it's going to make what it needs to make more of the cells and more of itself, because that's what viruses like to do. Okay, no problem. You would have some extra growth, and like a wart or a benign growth might form as a result of this. In fact, often does. But once in a while, what happens is that these chromosomes from the virus, they end up getting integrated into the host DNA. Now, in some cases, that also might not cause a problem. I mean, if you integrate it into some junk DNA portion of your chromosome, whatever. But what if you integrate this into a region that's highly replicated or it has a high rate of transcription and translation, and you're making a ton of protein? Now all of a sudden you've put things into overdrive. Or what if it's interfering where it integrates with a gene that's a cell cycle checkpoint regulator or something? Now you've caused problems there. So sometimes integrating this type of DNA from your virus can indirectly lead to massive growth, and now you can have a tumor that can become malignant. So before I tell you more about that, I'm going to pull up a couple of slides as a review for you. So that's why I have this written here. You've already seen this information. Remember, we talked about DNA damage and how you can try to repair that damage with this pathway that's p53 dependent, and that you can halt the progression of the cell cycle by inactivating CDK-based complexes. This is what we talked about not that long ago in the cell cycle and apoptosis lecture. And we also discussed how apoptotic proteins, how important they are, including how p53, in addition to being a part of this pathway, is also a regulator of cell apoptosis when necessary, and can turn on various pro-apoptotic genes and can inhibit anti-apoptotic genes. So bear that in mind. And then the second thing I've shown you before I want to pull up real quickly is this retinoblastoma protein, which again, normally when you want a cell to divide and you have a mitogen that's activating a receptor and stimulating mitosis, one of the downstream effects is going to be to phosphorylate and inactivate retinoblastoma, which then allows various genes to be turned on based on this green thing that was being held by retinoblastoma, being able to now bind to DNA, and it's a transcription factor that can then activate transcription. So retinoblastoma protein and p53, two proteins that are really important, right? So if we go back to our human papillomavirus, what happens actually is that when you have this virus, it makes many proteins, and these are two of them called E6 and E7. Well, here are two ways it creates problems for the host. The E6 protein binds to p53 and destroys it. All right, so now you have this p53 protein in your host cell that you've infected that would have helped kill off the cell because of an infection, and whoa, what's going on here? I'm being told to divide. Kill off the protein. Now the cell has lost one of its main defense mechanisms and can't trigger apoptosis. So apoptosis is disrupted. And that checkpoint also, remember, here is disrupted, the G1S checkpoint. So in two ways, by hitting one protein, you've caused problems for your host cell. E7 protein binds to retinoblastoma protein. And when it binds to it, it actually changes its conformation, phosphorylates the protein basically, or makes it look like it's phosphorylated, I should say, so that it looks like this, lets go of this guy, and now cell proliferation can move forward. So this is mechanistically how a virus is being really sneaky, and its goal is simply to make more of itself. How do you make more of itself? You make more of the host that you're in, so that it makes more of you. But if you take this out of control, now you're at risk for cancer because you have now hit multiple different important proteins that regulate apoptosis and that regulates the cell cycle. So, huge problem, right? Any questions about that? Okay. All right. So, this is just a nice overview slide, really simple slide of talking about what happens. That normally you'd have cells dividing, you'd have a damaged cell, and it would apoptose. But in this case, with cancer cells, they just keep on going because some of these checkpoints have been co-opted or destroyed or inhibited. So again, to reiterate a big point that I keep making, cancers then arise from violations of the basic rules of cellular social behaviors. When good cells go bad, normal cells divide only when they're needed, and then they don't when they're not. Normal cells live when they're needed, and they die when they need to die. And normal cells are good neighbors. They do their job, whatever it is, in a multicellular organism. They stay in their place, and they don't move into other territories or neighborhoods. Whereas cancer cells ignore all of that. They proliferate when they're not supposed to, they don't die when they're supposed to, and they move into territories where they're not supposed to be, and that's invasion and metastasis, which we'll get to next lecture. So, getting back to your question about seeing your physician, Cancer cells often proliferate indefinitely, but unfortunately, we have a hard time detecting tumors until they have hundreds of millions of cells. That's just a technology-based issue. How do you know, looking at me, what's growing inside of me? And do we have the technology and the cost and the means to go in and really carefully look for those things all the time? That's maybe the future, right? 100 years from now, where you can have some sort of really artificial intelligence-based scanning mechanism, Star Trek style, that's able to just kind of look at you and detect really tiny tumors and stop them maybe with treatment before they can get out of control. We can't do that right now. And there is a lot of research trying to figure out how we can better detect tumors at an earlier stage. So that's kind of shown here in population doublings, number of times the tumor cell population has doubled, and the change in diameter roughly of the tumor. And this is a logarithmic scale, right? So 0.1 millimeters wide, 1, 10, 100 millimeters wide. Notice that your tumor is first visible on an X-ray. That's if you X-rayed it and saw it here, when it's already got 10 to the 8th cells. That's a lot of cells. So it needs to be this big to really see. Think about all that time that it was undetected. The tumor is first palpable at 10 to the 9th, and notice how little there is time for growth before often, not always, but often at 10 to the 12th cells, the patient can die. So one of the things that's really important moving forward, and maybe some of you can contribute to this research, is how do we increase this time? How do we find these things sooner and give people more of a chance for treatment, right? Something to think about. Okay, so here's a really important point that you've kind of heard me talk about, but I wanted it to be really explicit. Cancer is developed by an accumulation of mutations. If I mutate just my p53 protein, I'm not going to get cancer. There are lots of different backup mechanisms and checkpoints and this and that. You know, it would be a problem, but not a cancer-causing problem. What happens in cancer is that you have multiple mutations that strike multiple critical components, and then combined, you can end up with cancer. This, again, is why we tend not to get it until we are much older, because it takes many, many years of cell divisions and mistakes and not catching them to get enough mutations in critical genes to end up putting you at risk. All right? And cancer cells evolve, which gives them an increasingly competitive advantage. So let's talk about these two things in a little bit more detail. So let's say you've got a cell sitting here in this is an epithelial layer growing on top of a basal lamina. So you could pretend it's, you know, the outside of most of your organs or your skin, whatever it might be. And a cell gets a mutation for whatever reason that increases its ability to divide like crazy. Now, it's not cancer, but it's dividing faster than its nearby cells are. So as time passes, its progeny, the cells that came from that first cell, which have that mutation that it acquired, are going to be an increasing percentage of the cells here, right? Now let's say you get a second mutation, and because you're dividing more often, it's kind of a runaway train thing, right? Because now you're dividing more often already from the first mutation, so you're now replicating your DNA more often, which means you're more likely to make mistakes more often, which means you're more likely to get mutations. And so now you have an increased risk of that second mutation, which then could increase your advantage. All right? Now, it could also decrease your advantage. It could kill the cell off. But in cases where it increases the advantage, now you're on further turbo boosted, if you will. And now maybe you get a third mutation. And that third mutation now gives the cell the ability to invade other tissues. At that point, if you acquire a mutation like that, and that's a big if, but if you do, that is the key now to getting away from your original location, metastasizing and spreading the cancer, and then therefore actually causing harm to you. All right? So, and that's kind of shown here now. This mass of cells, which was confined here, is now able to start moving away and go other places. So it really is a multi-step process. It requires multiple mutations. So now I'm going to act even stranger than I normally do in class. And to show you this, I'm going to actually show you a really neat video of bacteria growth as an analogy for what I just described to you here. And this is a cool video on its own, but I think it illustrates the evolution, the advantages of evolving different things with bacteria on a surface. So what you're going to see here in this video is that there's going to be increasing concentrations of an antibiotic, and they're going to explain that. And then they put some bacteria at the very beginning here and give them many days, and they just time-lapse this. They made a video over many, many days to see what happened. So yeah, check this out.
We ended up building was basically a petri dish. except that it's 2. And the way we set it up, so what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Okay, so really interesting and important to think about in terms of antibiotics.
Fighting's not that easy.
And the overuse of antibiotics that we have right now in our clinical treatment on its own, right? I mean, wow, how quickly that can happen in, what was it, 11 days or something like that? But additionally, an analogy for what's happening with these cancer cells, where if you get the right mutations over time, you are at an advantage and you keep moving forward, sort of past these barriers that have been put in place by your intrinsic biology to stop cancer from forming. So cell cycle checkpoints and apoptosis promotion, all those things, if you think of them as barriers in that video, cancer cells are going past those barriers because they're evolving mutations that take them past. And actually, I should say normal cells that are becoming cancerous, because then you become cancerous once you've gone past enough of those barriers. So just something to think about. Okay. And this is actually shown in this image here, right? I mean, it's the same idea. You have these environmental constraints that are virtual barriers, and a cancer founder cell that could become cancerous gets a mutation, and then maybe one of them gets a second and a third, and so on. So that's kind of what we just went over. Okay. Now, mutations that produce cancer must be just the right type. Sort of a Goldilocks kind of a thing, an analogy I've used before. Not too much, not too little. Because in a normal situation where you've got your normal cells and you don't have a bunch of mutations, you're not going to be cancerous by definition. If you have too many mutations, so that you have crazy amounts of genetic instability and nothing's working, you're just going to die because enough things will be triggered to cause apoptosis to happen. Or the cell won't even be able to divide enough to be a problem. So you really have to have this just right in-between level, where you had a couple of mutations or a few mutations in critical genes so that you can still divide, and in fact you're being encouraged to divide more, and you can survive and actually go past these barriers to form cancer. So what are the actual genes that are most often affected? You can kind of guess because we've talked about the processes. They tend to be genes that are important for cell cycle, for apoptosis, for growth, all that kind of stuff. So there are two main classes of genes, and these are really sort of more clinically oriented definitions, but they're important definitions, oncogenes and tumor suppressor genes, and that's what we're going to talk about next. All right, so cancer critical genes often function in normal cells to promote or inhibit survival and or proliferation. What I've been telling you, right? They're just genes doing their job, the proteins are doing their job, But if you get mutated, you could have a problem. What's shown up here, let's first look at the top part, A, is where you can get what's called a dominant mutation. Now, hopefully you remember this from genetics, or you're taking genetics now. Loss of function, gain of function, dominant, recessive, all that good stuff applies here. All right? So when you have a dominant mutation, so when you have a dominant mutation, you have a mutation, you have two copies of every gene in your DNA. A dominant mutation is one where if you had one copy of the two mutated, that's sufficient to cause a problem because of any number of downstream reasons. And usually, for example, let's say you're making too much of the protein, and now it's a problem because even though you have the normal gene making normal protein, you just have too much protein. So that's one example of how that happens. And we'll cover a couple of examples. So when you have a dominant mutation in what's called a proto-oncogene, that then creates what's called an oncogene. Now, I want to be really clear about a point that gets kind of confusing. What is a proto-oncogene? It is something that we have chosen, we being whoever named this stuff, to name a normal gene that's doing a normal thing, but that is known to be a potential risk factor for cancer if it acquires a dominant mutation. In other words, a proto-oncogene, proto for like early or before, a proto-oncogene is just a gene, right? So it's just a gene that's doing its job, but it's called a proto-oncogene as a clinical term, if you will, or a definition, a biological definition, because what it means is that if it's mutated in a certain way, it can become a danger. All right, that's why the proto is there. So I want to make that really clear because it can get confusing sometimes for folks. Well, what's a proto-oncogene? It's just a gene that if mutated, could become an oncogene. If you get a mutation that's a dominant mutation that increases its function, then you have what's called an oncogene. Because having that increased function, what it does in the context of cancer, and in the examples we're talking about, is to stimulate cell survival, proliferation, or both. In other words, you've done something to boost the protein level or function, or even, well, let's leave it at that for now. But whatever you've done, that single mutation has increased the cell survival, increased the cell proliferation, or both. And as we know, that is going to put you at risk for cancer. On its own, it does not cause cancer, but it puts you at that risk, right? So you get excessive cell survival and proliferation. Okay? Recessive mutations down here, different animal entirely. Recessive loss of function means that if I mutated just one, like shown here, no problem, because the other copy in this example down here will do the job, and that's shown here. So one mutation, no problem, this is not working, but the cell can make its RNA and protein from here and keep doing what it needs to do. But if you get a second mutation in that same protein, in that same gene, and now you inactivate that gene copy, now you don't have it anymore, or at least not a normal version of it. Now in the absence of any of it, you have a problem. So it's a recessive mutation, requires both copies to be mutated, and doing that eliminates the function of the gene. And then look at the end result, promoting cell survival and proliferation. Promoting cell survival, proliferation. The end result, in terms of what the physiology is the same, but you're just coming at it a different way. And so in this case, these are called tumor suppressor genes, because their normal function is to do something that stops cancer from forming. For example, P53. P53 is important for cell apoptosis, right? We've covered that extensively. If I got rid of one copy of P53, the other one could still do its job. If I got rid of both copies of P53, who's going to start the apoptotic pathway, right? And now you've gotten rid of a tumor suppressor because you got rid of both copies. So these terms, oncogene and tumor suppressor gene, are names given by people to a very simple concept of dominant and recessive mutations that are relevant to cancer. We get mutations all the time. So if you had a dominant mutation that wasn't relevant to causing cancer, like if I mutated a gene that turned me here from brown to, I don't know, white or black or something, that is a mutation, but it's not causing cancer. So it's not relevant to being an oncogene or a tumor suppressor gene, right? If I get a mutation that suddenly made me really tall, which would be awesome, since I'm really short, Okay, fine, but if it's not relevant to causing cancer, then I could finally play basketball, which I love to play and I'm terrible at, but it's not going to cause cancer, so it can be a dominant or a recessive mutation, but it's not an oncogene or a tumor suppressor gene. All right? So these are very cancer-specific definitions based on the mutations that you have. So what we're going to do next is look at these in more detail. The first one we're going to look at is dominant mutations, so oncogenes. So this is the same exact figure that you just saw. And here's some information that repeats what I've already told you. Oncogenes cause cancer in a dominant fashion. They arise from normal cellular genes. Remember, they're called proto-oncogenes. That just means it's a normal gene. Or they're introduced by viruses. Kind of like what we talked about with the human papilloma virus, where you can introduce stuff into the DNA. They tend to be signaling pathway components. That makes sense. We know signaling pathways are so important for everything we've talked about. And they code for abnormal forms or excessive quantities of such proteins. So there's two different things listed here. Either you have a funky, different mutated protein, or you just have a normal protein but way too much of it. And either way, the effect is doing too much of something in this example. And then the end result, again, we've covered it, excess cell proliferation and or cell survival, inhibition of apoptosis. These are some examples of how that happens. So how do you go from a proto-oncogene to an oncogene? So let's say you've got a gene here in blue in the middle of all your DNA. One way that's going to happen is you take your normal gene, the proto-oncogene, and you move it to a new place in the genome. It gets picked up, right? A mutation happens or an error in copying your DNA or whatever, and you end up picking this piece up and putting it somewhere else where there's a different promoter driving its expression at a higher level. So the gene's fine, but now it's being made at, say, 10 times the amount it should be made because it's in a different place. And now you get normal protein in excess. Another example would be that you just ended up accidentally making too many copies of your gene. Now, if each of these is making the same amount of protein, you get an extra protein again. Another possibility would be that it stays where it is, and you still have just one copy, but you end up mutating something in the control mechanism here, the promoter for this gene, and it just turns on more of itself. So that makes more RNA, more protein. And then finally, you could have a mutation within the gene itself that makes it, let's say, extra active or more likely to not be degraded by, let's say, ubiquitination and degradation in the proteasome or something like that. So the end result, of course, in all of these cases is there's an increase in the function of the gene and the protein that it makes. Notice that these are 4 examples, and only one actually had a mutation in the gene itself. So it illustrates the diversity of how this can happen. It's not just that you have a gene that's important and you mutate the gene. That's one of the ways to do it. But look at all the other ways. Mutate the control mechanism, make more of it, move it somewhere else. It's an imperfect process inside of our cells. And when it screws up in one of these different ways, you can turn a proto-oncogene into an oncogene. This slide is the exact same thing from your book, shown a different way. I give you both because I like this one better. I think it's a better one, but this is actually from your book, so whatever you prefer. But it's saying the exact same thing, mutations in the gene, amplifying the gene, rearranging the chromosomes, exactly what we just went through, all right? So take a look at whichever one you like better. Yes?
So maybe because these mutations do not be visible is like the errors in endonuclease and hexanuclease, so it skips it and doesn't see it?
Yeah, so that's you're thinking ahead. That's a good question. So the question is, would you have errors in, let's say, endonucleases or exonucleases that would then result in your DNA repair machinery not seeing these mistakes? That can absolutely happen, and we'll actually get to that briefly a little bit later. That's one thing that can happen, is that you end up, your DNA repair machinery starts to break down. In fact, it's one of the most common ways because it's, the analogy I like to use is, if you think of these things as different machines in a factory, different proteins, and a machine breaks down, you could try to repair it and get your factory running again. But if you're by using, let's say you have a machine that repairs those machines. But if your machine that does the repairs breaks down, now you can't repair anything. And that DNA repair pathway is like that. Because your DNA repair pathways are repairing all of these different things, if that breaks down, then any mutations that happen or that are hard to correct, you get a more rapid accumulation. So yes, that can and does happen, but it's not the only thing that can happen. Oftentimes, it can be as simple as it missed the mutation. So your DNA repair, your proofreading that the nucleases do, it's not perfect. It has an error rate. And that error rate is low. I don't remember if it's like 1 in a million bases or something like that, or 1 in a few. Do you remember, Joe? No, I don't know. It's really low, but think about how many bases of DNA you have. So over time and over dividing and dividing millions of times, even at a low rate, you're going to start to miss a few things. Yeah. Any other questions? Yeah. Yeah, so that's a good question. Could we target the oncogenes in cells for cancer treatment, or would that just affect every cell? Sort of the answer to both questions is yes, to a point. So for example, one of you actually asked me about this after last class. You may recall way back when we talked about a drug called Taxol that can inhibit that causes problems with microtubule rearrangements and the dynamic instability. Microtubules are important for cell division, or for mitosis and moving your DNA apart and all that good stuff, right, in the cell cycle. So you can inhibit cell proliferation by using a drug like Taxol. And what you're doing is you're targeting, it's going to hurt all of your cells, which is why chemotherapy is so tough on people. You're poisoning your body. But in that example, the idea is that your cancer cells are dividing more often than your regular cells. So you'll disproportionately affect those dividing cancerous cells more. So that's why it's used as a treatment. Plus the fact that if you're in bad shape because you have cancer, it's just worth the cost to try and do the treatment to treat the cancer, even if it damages the body. And then hopefully you remove the cancer and you recover from it. So that's one example where you try to target a general process that might, because that cell proliferation that's happening in those cancer cells is likely due to mutations in these various genes. Now, that doesn't directly address your question, but that's an example of taking a general process and treating it, even because it is more prevalent in cancer cells. To more directly address your question, yes, some of the proteins that we're discussing, folks are doing tons of research on trying to come up with better ways to, let's say, decrease a protein that too much of it is being made, or inhibit its function because it's helping to promote cells to proliferate. And I think we'll cover some examples that might touch on that. But yeah, it's definitely an important area of targeting treatment. Yeah. Yes?
I was wondering if the normal cells ever react to the cancer cells and causing any apoptosis?
Yep, super awesome question as well. Do the normal cells react to the cancer cells by causing apoptosis? Right. So that's a great. You've done me a favor by, it's a throwback, hashtag throwback to what I taught you guys a while back, not that long ago, about the extrinsic apoptotic pathway, right? The one where cells come in and they trigger apoptosis. Normally, if a cell is getting out of control, you're going to want your normal cells, your immune system, to come in and signal that apoptosis. And absolutely, that happens. In fact, we probably end up with precancerous cells or cells at risk of cancer all the time, but we just never see the vast majority of them because your immune system, your defense mechanisms, whether it's the extrinsic apoptotic pathway or just DNA repair mechanisms or apoptosis inside of the cell, eliminates the threat. So yes, your normal cells will try to kill off the cancer cells. The problems happen in two major ways. One is that cancer cells sometimes evolve mutations that help them shield the problem from being detected. So if, for example, you're out of control and you're secreting something that lets the nearby cells know that this is out of control, what if you stopped secreting that molecule or those molecules? Then it'd be hard for the nearby immune system cells to know that they should kill you off. That's one way that it's a problem. A second way that the problem develops is if the cancer cells or the precancer cells are dividing so rapidly that even if there's a partial immune response, they can't keep up with it anymore. Or let's say that a few cells snuck away and metastasized, right? So in a patient who has cancer, your immune system is, in general, fighting that cancer. But if the cancer spreads so quickly and so broadly that you can't keep up with it, or that it can hide out in places where maybe immune system cells can't even see it, that can cause a huge problem.
Do the healthy cells also ever try to actually
Yes, so your normal immune system is constantly trying to induce apoptosis in cells that, for a variety of reasons, need to undergo apoptosis. Yes, absolutely. Yeah. Any other questions? Okay. Okay, so here's another review slide, if you will. This is the GTPase RAS that we covered in some detail. And some of you, this was a tricky test question for you, right? So this is a good review slide for that. RAS, remember, is a GTPase, and that it's turned on, this green thing is turned on when you remove the GDP and put the GTP in its place, and that's stimulated by a protein called RAS-GAF, shown here in blue. So what you might recall from our discussion about this is in the next slide, that when you have activated RAS protein, you end up activating this whole kinase cascade, which we're not going to go through right now, but that results in mitogen-activated protein kinase and other molecules being activated. RAS is really important for cell proliferation. So when you have such a critical component that's so important for cell proliferation, based on what I've already told you, you can imagine it's a risk. If this one was mutated, that could put you at risk of cancers, and in fact that is the case. So if you again remember review that you can have mutants that can't hydrolyze GTP. If you're a GTPase, the RAS, and you can't act as a GTPase and hydrolyze your GTP to turn yourself off, then you're always staying on. And if you're always staying on, you're always signaling. And if you're always signaling, then you're going to have proliferation downstream of that. And it turns out that mutations or RAS is always on are associated with cancer, quite often. Uncontrolled proliferation, even in the absence of mitogens. So here's a little, practice question for you then. Is RAS an oncogene or a proto-oncogene, whatever you want to call it, or is it a tumor suppressor? Raise your hand if you think it's a proto-oncogene. And raise your hand if you think it's a tumor suppressor gene. All right, cool. So not too many hands, there are a lot of hands that never went up at all, but I see that the hands that went up for oncogene, proto-oncogene, so that's great. That is the correct answer. This is a case where you have a gain-of-function mutation that is causing the protein to always stay on. In other words, you have more activity of the protein. You're doing more of something, and doing more of something is leading to more proliferation. That's exactly what I showed you in those earlier slides about dominant mutations in proto-oncogenes, making them into oncogenes. So that's great that you guys are getting that sense already that this is an oncogene or a proto-oncogene. Okay, here's another one. Remember that receptor tyrosine kinases activate AKT. And that AKT, this was also on your exam, leads to cell survival being promoted by this survival signal because it, excuse me, let me just. I'm not sick anymore, but it's just a cough lingers. Okay. I'm not going to get anybody sick, I promise. Right. So the survival signal is separating these components out, and it's promoting cell survival because you're inhibiting apoptosis. And so that's another example where you can have mutations. If you always had the survival signal on, you would be promoting cell survival, right? So it's just another example of a case where you can have a proto-oncogene putting you at risk. In this case, you could view any of these components that are, let's say you had overactive AKT, you could consider that an oncogene, right? A proto-oncogene becoming an oncogene. Or if you had overactive BCL2, right? That would be an example of a proto-oncogene becoming an oncogene. All right, so that covers proto-oncogenes and oncogenes. Any questions about that before we get into tumor suppressors that I haven't already answered? Okay. So then let's go back to tumor suppressor genes, recessive mutations, loss of function, what I showed you at the beginning, right? 2 copies inactivated, you have a problem. You have now excessive cell survival and or proliferation. And the name really is appropriate, right? Tumor suppressor gene. One of its jobs, not because it's trying to suppress tumors, just in its day-to-day job. Its job is to promote apoptosis or to inhibit cell cycle progression, to be a checkpoint, and all those things that help things take a pause instead of dividing And so by doing that, you're inhibiting tumors from forming. And if you get rid of it, then you have a problem. So their normal function is usually to restrain proliferation. Some that we're going to discuss, in fact, I think the only ones we're going to discuss in the next few slides until we run out of time today, are the retinoblastoma gene, which you've already heard about. Remember, that one restricts passage and is a restriction point for the cell cycle. P53, which you've heard so much about, and APC, adenomatous polyposis coli, which inhibits the Wnt signaling pathway by degrading beta-catenin, and we'll get to that eventually, depending on time. All right, so just like I had for the dominant mutations, these are 4 examples of how you could get to having both copies lost in this recessive mutation situation. So you've got your 2 copies here, and let's say you had a normal gene here, and then for some reason you got a mutation here. So you've got 1 mutation, no problem so far, but now you could get the second one lost or mutated. And so these are 4 very generic examples. You could lose the whole chromosome, and then all you've got left is your messed up copy. You could have the region of the gene deleted. You could have an actual mutation in that gene. Or you could have the gene activity being silenced by some other sorts of epigenetic changes. So again, very broad ideas here. The end result is that you end up with 2 copies of your gene that are not functional. So let's talk about retinoblastoma. This is retinoblastoma. It's named retinoblastoma because it occurs in the retina of the eye. The retina of your eye is one of those tissues, just like the inside of your nose or your mouth, that are pretty exposed to your environment. Of course, we look at stuff all the time, and we have to take light in, including UV light. And so the tissue in your retina is particularly susceptible to accumulating mutations because of UV light-based mutation of DNA. Now, most of the time, this is not a problem until you get really old. As you know, folks, when you get old, you can have problems in your eyes. But usually, it's not a problem in a normal, healthy individual because we're doing what we're doing. So in a normal, healthy individual, if you had 2 normal copies of the RB or retinoblastoma gene, which makes the RB protein that I told you about, you're not going to have a problem. However, one of the reasons that kids often get retinoblastoma is that a mutation in the RB gene is often passed along from parents to their kids. And when I say often, I don't mean all the time. I just mean compared to other inherited mutations. It's one of the more commonly mutated genes that can be passed along. And a discussion of why that is sort of, we don't have time for, but if anybody's interested, we can talk about it after class or something. So you pass along a mutation of this gene, right, as shown by this X in 1 copy of the chromosome. Still no problem because you've got that second copy that's active and doing what it needs to do. Remember, this is a cell cycle checkpoint gene. So is this, well, I guess you know the answer because it's in this section, but this is a tumor suppressor gene, right? You have to get rid of both copies of it to have a problem. So in this example here, these cells are just fine. But once you have that 1 copy mutated, now you have a risk because it's your eye and it's exposed to those sources of radiation that maybe during your lifespan you will acquire that second mutation at some point. And that can lead to the excessive cell proliferation that can lead to neuroblastoma. So as a result of this, most people who inherit a single mutation, they don't have any problems at birth and early in life, but at some point you are then likely to actually get retinoblastoma because you are likely to inherit that, to develop that second mutation. Very rarely, and you have non-hereditary retinoblastoma. This is just pure bad luck, where you have a normal cell, it's dividing, and then at some point you accumulate mutations in both copies of that gene. And then now you've got a problem, obviously, because you're at that same situation that you have right here. So excess cell proliferation. Because this is so rare, only about 1 in 30,000 people develop a retinoblastoma tumor throughout their whole life. So it's very rare to get that happening randomly, right? All right, so that's the retinoblastoma gene, and an example of a tumor suppressor that's a cell cycle checkpoint protein that, if mutated, causes problems. All right. P53. So P53, you've heard so much about, is another example of a tumor suppressor gene. And I just want to share with you this example. It's out of date. This is pretty old data. I forget what year. But the general point is very much salient today. This is the distribution of various cancers in developing and developed countries and the incidence of p53 mutations in those cancers. Now, before we look at this, I want to remind you the p53 mutation on its own does not cause the cancer. But what this is saying is that, so let's say in lung cancer, 70% of those lung cancers have a p53 mutation, as well as a bunch of other mutations. Okay? So look at the prevalence. As you can see across various cancers, high percentages have p53 mutations. All right? So, and we won't get into the differences between developing and developed countries. That's an epidemiology kind of thing. If you're interested, you can look at it and speculate about those differences, diet and behaviors and so on and so forth. But the point is that p53 is a really critical aspect of developing cancer if you have both copies mutated. And as a reminder, this is what happens when you damage your DNA. I don't expect you to memorize this pathway here, but the idea is what I've told you before, that you have p53 being incredibly important both for cell cycle arrest, the DNA, the cell cycle pathway, or you remember we talked about this, and I expect you to know this, that p21 is activated by p53. This is all on that slide I put up earlier, that you can promote cell cycle arrest to avoid going from the G1 to the S phase. And also we've covered in detail that p53 is responsible for apoptosis. So again, critically important aspect of everything we've talked about is these types of tumor suppressor genes. So let's see, I think I had some cool videos on Twitter to show you very quickly. Probably a good time for a science Twitter break. This is a macrophage. I can see people are kind of fading a bit. So macrophage killed by T cells and eaten by another macrophage. This is kind of just fun because, remember, I didn't show you an apoptosis video, I think. So let me just play it from the beginning. These are T cells, part of your immune system. These are macrophages. And then we'll see what happens here. Keep your eyes on this macrophage. So the T cells, you can imagine, they're responding to signals right now. There are signals that these cells are detecting that tell it there's something wrong, and they're inducing the extrinsic apoptotic pathway. And now you can see this cell starts to bleb up and start to look really messed up. It's going to die here as it falls apart because it was told to die by these T cells. And once their job is done, they take off and they go check out other cells. They're sort of checking in on everybody and making sure that everything's fine. And then this macrophage does its job as a macrophage. It says, oh, dead cell, I'll go ahead and suck it up, clear the debris, and recycle it and use it for whatever. So isn't that cool? Bits of cellular stuff being transported and sucked into the cell as it does its job. So that's kind of cool. All right. This one I showed you a long time ago, and I just wanted to show it to you again because now you're learning about cancer. T cells killing cancer cells. So here's another T cell. It's going to come in. The fluorescence you see is because of an indicator that somebody put in. This lights up, and this T cell binds to it and undergoes apoptosis. So, examples of the things you've been learning about. And we won't cover this one today because I haven't told you about apoptosis yet. So with that, we are going to pause the lecture at this point. You have some extra credits that we do, and your TA is going to hand out extra credit now. Don't bother texting your friends. We're not going to, people run here, it's too late. You're going to get them now, and you have to do it now and hand it in before you leave. And then that will be part of your extra credit for the class. So good for you for attending today. You get to benefit from that. And then once you're done with your extra credit, you can take off. If you didn't get one, just raise your hand and we'll bring one to you. If you have extras, just put them aside, leave them, it doesn't matter. And again, you have to hand these to your TA before you leave for it to be graded. We won't be taking anything after 12:15. All right, looks like everybody's got one. Also, in addition to my candy today, there are leftover donuts that the last instructor told me about, so grab a donut if you want one. They might be a day old, but they look good.